absolutely exhilarating talk. Thank you so much to the organizers for inviting me to speak, and especially for accommodating me at a distance. I would have dearly loved to have been there, but it did not seem like a good idea to travel from Australia at this particular time of time, and indeed it has so proved, because we have strikes of ground staff and all sorts of things at our airport. So it's a good thing I didn't come. But this has been very, very stimulating. And both of the talks so far have been, have addressed structural aspects of research. And I am going to move us in the direction of more theoretical principles. I don't have any gorgeous slides to show of empirical work that I'm doing, because I do all of my work in my head. And so in rough outline, what I'm going to talk about are 7 mechanisms which appear to be conserved in cognitively generated behavior from bacteria to humans, which is a pretty hefty thing to say. And I am emboldened in this project by the principles of neural design set out by Peter Sterling and Simon Laughlin in their wonderful book of 2015. Before I get into that, I would like to say a few words about how I got here, simply because, it's like, it seems like I just decided that bacteria were cognitive or something like that, and went out to demonstrate it. And that really couldn't be the farther from the truth. I had a quandary when I was doing my PhD research in not even a tangentially related field. And that was that I needed to answer the question, what is cognition, before I could do anything on the topic that I had chosen. And my supervisor very kindly allowed me to wade into this swamp. And I'm glad he did. When you ask the question, what is cognition? It raises 3 questions right off the bat. What is it? What does it do? How does it work? When I asked the question, what is it, at the time that I was doing this work, the only answer seemed to be, it had to do with the human brain. It was always an advert to the organ itself. And this I found quite confusing, because the organ regulates the entire body, not just the cognitive parts of the body. And it seemed like it was going to be a fool's errand to completely tease apart the physiological metabolic aspects of what the brain was doing and the cognitive aspects that the brain was doing. So I didn't think pointing to the brain was going to be very helpful in answering these 3 questions, which were my quarry. When I asked, what does it do? I was told information processing, which is true enough, computation, and representation. I could understand the first, and I could understand the computation might be a way of describing what it does, but to say that cognition is computation, and moreover that it's computation over representations, which are these obscure entities that we identify in our first person experience as somehow being manipulated when we think, that didn't seem like a very safe foundation to be approaching this question from. And when I asked, how does it work? I was not very surprised to learn we're working on it. It's really, really hard. So my burning question at this point was, where's biology in all of this? Because at that time, it wasn't anywhere. I actually had a university lecturer tell me, I asked, where's the body in all this? Where's emotion and motivation in all this? And he said, we don't need those because we can abstract away from them. And I didn't believe that. And I couldn't understand why nobody was talking about evolution, ecology, autopoiesis, thermodynamic constraints, adaptation, genetics, development, goal orientation, real world behavior. And nobody at that point was really attempting animal comparisons except with primates. And even then, the bar was set so high for primate behavior. It was almost like, well, if you weren't sort of a mini human or something like a human, you weren't going to get anywhere. And that didn't make any sense to me at all. And then I had the very good fortune to read the last two sentences of a book that never made much sense to me anyway, by Jerry Fodor, who was one of the celestials of cognitive science from the late 1960s until he wrote this book, really. And he, even though he was only a philosopher, he had advanced two hypotheses that had gained neuroscientific traction. The first one was the modularity of mind hypothesis, which was based on what seemed to be the regionalization of the brain at a structural level, which many of you are obviously familiar with, and which, even though it gets harder and harder and harder to pinpoint things in particular places in the human brain, it keeps hanging around like the ghost of Hamlet's father. The second one was, the second hypothesis was the language of thought hypothesis, that the mind, that thinking and mental activity was relevantly like words in a language. And then I read the last two sentences of this book. What our cognitive science has done so far is mostly to throw some light on how much dark there is. So far, what our cognitive science has found out about the mind is mostly that we don't know how it works. Understand, this was at the turn of the 21st century. But the first thing I realized was that I actually didn't have to look at the human brain first, and that it was a crazy place to look because it was the most complicated organ on the planet. So I immediately decided I didn't need computation. I was wrong there, but that's what I was thinking. And representation didn't seem to be pulling its weight, so I could ditch that. And of course, keeping information processing was, if you'll excuse the pun, a no-brainer. So what does biology tell us? First, it tells us three very, very simple things. Organisms seek to persist or survive. They are dynamically self-preserving. Second one is that organisms engage in action to facilitate their growth and thriving. And the third one is that they engage in actions to facilitate their reproduction. Very, very simple, very, very complicated. Because these three sort of implicit goals of life had all kinds of implications that I found were quite helpfully constraints on thinking about cognition. The first one was that the the organism is maintained far from thermodynamic equilibrium, that it is self-producing or autopoietic, which means that it has to interact with the milieu to extract matter and energy. And because this is a very complex process, and energy resources have to be distributed intelligently and efficiently, that meant that there had to be control mechanisms and regulatory mechanisms. That also meant that there was continual adaptation to the surroundings and a changing internal state. And again, in deference to the no-brainer, it was obviously clear that adaptive behavior depends on information. But that meant that external and internal monitoring would both be needed. And it also meant that there had to be a means of relating external and internal information. And on top of that, it meant that there had to be a means of valuing information as acceptable, harmful, or advantageous. Because if those things can't be determined, then there isn't much for the genome to do, really. Except, organisms are not wind-up toys, as we all know. And because value depends on a multitude of factors, error is always possible, which means that there has to be a means for detecting error and making adjustments. And these points, these further elaborations of the three core existential goals, also fed back in the reverse direction. And what they provided was a framework for looking at how we have sensing, how we have memory, valence, learning, decision-making, anticipation, and communication, which all turned out to be a cognitive toolkit that now I have come to believe is possessed by every organism that is alive. Now, all of those elements of the cognitive toolkit can be found in bacteria, and I will rabbit on mostly about bacteria in this presentation. And the question is why? And that is because, to me, bacteria are the bellwether. If you can find a capacity in bacteria, it has got to be and I'm talking capacities here, I'm not talking mechanisms or structures. It has to be in other organisms. And that led me to an approach which I call basal cognition. Basal here does not refer to the type of cognition that I'm investigating. It is the type of organism in which I am investigating it. And that includes mostly aneural organisms and those with very, very simple nervous systems. And I look very much forward to looking more deeply into the work on Platinaris. Fabulous. And from basal cognition, as an approach, it involves three approaches that are really but had not been applied in cognitive science to that time. This was the turn of the 21st century, and it wasn't until the latter part of the first decade that serious comparisons of cognizers were being made. And that is to study the simplest organisms that can display the cognitive phenomenon of interest, identify principles, and then scale up to more complex organisms and observe where the similarities and differences actually lie, not simply where we think they must lie. My proof of concept came with the work into the ion channel. Hodgkins and Huxley had set the challenge on the model of how ion channels operate in the 1950s. Roderick McKinnon, a biophysicist, was absolutely on fire to figure out how they worked. And he had developed an extremely elegant assay for identifying the ion trapping amino acids in the ion channel protein using scorpion venom. And so he, so he went off feverishly looking in Drosophila, the fruit fly, and found very quickly that he could not synthesize the amounts of the protein that he needed to do the work in that model organism. At about that time, it was discovered that bacteria had ion channels, and so he switched from the fruit fly to the analog in Streptomyces lividans. Now, the thing he found, and really surprised him, but then again, he reconsidered it and thought, well, duh, this is biology. Bacterial ion channels were equally sensitive to the toxin as were Drosophila ion channels, as were mammalian ion channels, which indicated to him that there was a structural and a functional similarity. But at that time, bacterial, the function of bacterial ion channels was not known. And it wasn't until several decades after McKinnon's work, oh, needless to say, he won a Nobel Prize for this work. But a couple of decades after he had done the work that he was received a gong for, it was only then that somebody, the man right underneath him, who is Gurul Suel at UCSD, started looking at what bacterial ion channels actually do. And what he found was that they have the same functions in bacteria as they do in neurons, and that's to signal rapidly over long distances and large numbers of cells. So after fossicking in sort of like the undergrowth of the evolutionary tree for many years, in around 2018, which is when I had the good fortune to meet Detlev for the first time, I was beginning to think that there seemed to be some common mechanisms that underlay cognitive capacity, and they were becoming clearer and clearer to me. And when I read this book, The Principles of Neural Design by Peter Sterling and Simon Laughlin, I became quite convinced just by their chapter 2 that that this was looking for those sorts of principles would be useful, even though I can't say that what I'm looking at right now are principles, but rather that the principles of neural design seem to dovetail with them quite neatly. So I'm going to tell you what they are here: sensing and memory, network activity, staying together, and cell adhesion. Remember, this is all at the bacterial level. Cell-cell communication, divisions of labor, which I'm definitely going to take your evolutionary unit idea on board, which is going to be problematic, but I think that that's the way to go. Oscillations and coordinated responses. So what I'm going to do now is I'm going to try and get through all 7 mechanisms and at least point to the kind of principle that Sterling and Laughlin devised, and there are 7 of those too, which is really quite creepy. I didn't realize that until this week. And I'm not going to go into what their principles are, just I will summarize them briefly. Okay, so the first mechanism for me is sensing and memory, and the principles that apply the neural design principles that apply at this level are the first 2 of Peter and Simon's. The first one is compute at the cheapest level. That means using chemistry with components that are irreducibly small and maximally energy efficient. Be wary of costs of speed over distance because electrical signaling is very expensive. So basically what we're going to go through now are the basics of computing with chemistry. This is pretty much all Sterling and Laughlin. I'll provide you an example of ion channel mediated bioelectricity and memory in Bacillus subtilis, and we will make a short detour into associative learning in amoeba, because learning is, after all, long forms of memory. There are other things involved, but without memory, there is no learning of any kind. Okay, I don't want to go into too much detail about bacterial 2-component signaling because it's a huge field. But let's just look at the 2 components they're talking about are a membrane-bound kinase in this sensor kinase, which in this case is a histidine kinase, which autophosphorylates. It induces ATP mobilization in itself, and then it phosphorylates another protein that is called the response regulator. These terms are specific to bacteria. I don't know why, but they are. They developed in a silo, and they haven't been more widely used. They're in a sensor hybrid kinase. There are intervening steps that also phosphorylate, and including a specific phosphotransfer protein, once again ending up at the response regulator, which in bacteria is either regulating a flagellum, or it's connecting to a transcription factor. Okay, computing with chemistry. One of the neat features of computing with chemistry is that protein networks operate near the thermodynamic limit of energy efficiency. And just as a clarification, information is transmitted whenever a source's change in state registers as a change in state at the receiver. Seems pretty obvious, but it's worth repeating. But there were 3 key mechanisms that Peter and Simon thought were key to enabling information transduction in protein networks, in protein pathways. The first one is that they are thermodynamically stable 3-dimensional structures at their free energy minimum. Now, I'm not talking about Carl Friston free energy. I'm talking about biophysics free energy, which a state in which proteins seldom sit. But this 3D structure determines the protein's physical and chemical characteristics and the role in the cellular economy. And the protein's capacity, the key to all of this really is the protein's capacity upon binding to switch into another stable conformation, which may alter its receptivity to further binding, affinity positively or negatively and may catalyze further reactions. Okay, this conformation switching capacity is allostery, and it can serve to amplify or terminate a signal, reset the information processing mechanism, and as they put it, equips a single cell. Whoops, where am I here? I can't see my own screen because I'm looking at myself. Can I stop looking at myself? Would anybody mind if I turned off my video? Never mind. It's probably too thingy at this point. At any rate, they say it equips a single protein, because I still can't read it, to compute by operating like a finite state machine. Now, the most important thing to understand about finite state machines, which I don't really understand all that well, is that they were the cornerstone of the cognitive revolution in terms of being able to define, instantiate defined sequences of state changes, each triggered by a particular condition, such as the presence or absence of an input or a conjunction of inputs. And from this, Sterling and Laughlin conclude, in brief, simple chemical circuits are capable of implementing procedures used in analog electronics, namely amplify, oscillate, differentiate, and integrate, as well as taking logs and raising to powers. Chemical circuits support the arithmetic operations add, subtract, multiply, and divide. Small chemical circuits also have the ability to perform more complicated functions. Now, when I read that, I think I nearly fell off my chair because I can't take issue with what they've said. But they feel pretty strongly that this is the case. And if it is the case, we shouldn't be surprised that there is actually quite a lot of computational power going on in single-celled organisms. Okay, so here is an example of electrical signaling in a B subtilis biofilm. Now, this area in the central bit is where the biofilm first started. It gets very congested, it's very dense, and it can get nutrient deprived because all of these zigzaggy things at the periphery here are cells that are expanding the biofilm, so they're eating happily away, and not enough nutrients are diffusing to the center. So they basically put out a cry for help, and that is a metabolic trigger in a positive feedback loop that induces potassium release, leading to depolarization of neighboring cells. Now, the depolarization wave coordinates the metabolic status of the cells in the interior and on the periphery. So the cells on the periphery stop eating, and the nutrients diffuse to the center, and the cells at the core are no longer starved, and everybody's happy. And then, of course, at another time, they send another signal, and the cells at the periphery start munching again. Okay, now there's an interesting case of light-induced memory in B subtilis biofilm. All of this is work by Gurel Suel's lab at UCSD, which I just think he's doing astonishing work, and he's building a foundation that will serve us well for a very long time. Okay, so here you have your flower-like Bacillus subtilis biofilm, which, by the way, all those wrinkles are similar to the wrinkles in cortex in the brain. They increase the surface area for, in this case, for oxygenation. And they applied a light to the surface of the biofilm that was impeded in some way that was blocked in some way and had holes in it, sort of like a silk screen template. And what that led to was this biofilm with the UCSD logo of those cells that were depolarized or polarized, and all of the other cells that weren't exposed to it. And this memory of that exposure lasted for quite some time. Now, quickly in the associative learning in amoeba, what I'm going to do is I'm going to talk this up quite a lot, but obviously this has to be replicated and replicated and replicated and tried in other things. But this was a heroic effort at trying to establish that. This was a pilot program that was expanded to 3 species, wild type and free living, at 5 labs and in 3 countries, that involved the treating and tracking of over 2,000 individual cells that were treated individually and washed and pampered so that they wouldn't fall apart. The procedures in this study are just amazing. And there were 5 experimental treatments: the control, which was just normal feeding, the application of electric field, which showed that the cells naturally were attracted to the cathode. And then they were exposed to a chemotactic peptide, which is a molecular signal from one of their prey bacteria. It might have been E. coli, I can't remember. And then there was the induction was the simultaneous presentation of the electric field and the chemotactic peptide at the anode end, the side that they don't generally want to go to. And so the conditioning test was the electric field without the chemotactic peptide. And one of the things that's interesting about this conditioning test is that they measured a number of relational properties. They really went out of their way to measure response intensity, directionality, and persistence times, total distance traveled, directional ratio, and average speed. And in this, there were quite a few tables, as you can imagine, and graphs, but this one is, I think, puts it in starkest perspective, and that is that of the unconditioned cells, when they were exposed to the electric field, 96%, 97%, and 94% of the species all were gravitating toward the cathode. After conditioning, 75%, 82%, and 79% were gravitating toward the anode. I don't know what, that very little is known about amoeba behavior, actually. It's, a lot needs to be done for anybody to even say what the galvanotaxis means for them as an organism, but it's interesting. Okay, the second principle is network activity, which accords with the neural design principles of concentrating resources and minimizing wire. Bacteria do actually make wire. They make filaments and other kinds of protrusions, but that's not what we're talking about here. Okay, so we're going to be talking about signal integration in circuits in B. subtilis and E. coli, and then we're going to look at large chemotactic protein arrays or nanobrains, which is a conserved architecture in flagellated bacteria. And I'm thinking that this might be something worth looking at as perhaps the first chemosensory organ. So, and the third principle that applies here is the efficiencies that can be gained from prediction. So the fact that bacteria like E. coli and Salmonella did not respond to combinations of attractants and repellents in an additive way has been known since the 1970s. And these nonlinear responses were a surprise, and it still remains puzzling. That bacterial signaling cascades could work like networks or circuits was first suggested in 1995 by a Dutch-led team who were studying the faux operon, which is an extremely complex operon that has global implications for B. subtilis, and is instigated when in situations of limitation of phosphate, of a particular phosphate source. And it was then that and other pathways were elaborated in detail by Dennis Bray in 2009 using a parallel distributed processing framework in wetware. But he didn't have the information that came in the next decade, which is a real shame because heaven knows what he would have done with it. Okay, so we're looking at something called a nanobrain. This is a kind of a jocular shorthand for something that is normally called a large chemoreceptor array. It was first discovered in E. coli at the turn of the 21st century and now recognized as highly conserved architecture for receiving and integrating signals from the surroundings, specifically in bacteria with flagella, a particular kind of motility. And the nanobrain, I'm going to use that shorthand because it's only one word, is composed of several thousand chemoreceptor proteins, and sits at the leading pole of the cell. Leading, that means if it's traveling in that direction, that's where the nanobrain is, like a brain. And here we have the first real microscopic looks at what these things might be in Rhodobacter spheroides. In the top, you see them just the dark organ, and in the lower ones, you see the fluorescing proteins, the fluorescent tagged proteins, which is really interesting. And the nanobrain isn't just a numbers game, however. The structural elements are also highly complex. And so I don't waffle all over the place. I'm going to crib here. Okay. These two, this and this, E and Q, are examples of the protein structure that makes up the array. And one of the things that I find interesting, it's like honeycomb. The hexagonal structure, even though the architecture of some of these arrays can be quite different, the hexagonal packing is it seems to be the most stable and energy efficient of all possible structures for these things. Now these are methyl accepting receptors, unlike many in bacteria. And as a result of that, it has a certain degree of complexity. There are 5 ligands, different ligands that are sensed by these chemoreceptors, and each chemoreceptor is a triplet ligand binding structure, and each structure can bind with up to 2 different ligands and can be methylated on 4 different sites. Each triplet converges on a single protein, that's key A, that calculates the output, but key W is an intrinsic part of its structural integrity. A chemoreceptor protein array may be comprised of up to 10,000 units, which in combination with other inputs controls motility via the flagella. Now, motility by flagella doesn't seem like much. I mean, yeah, it's running and tumbling, but actually it's much more than that, and it's much more than a random walk. It's much more directed than a random walk. There are twiddles and fiddles that, and this is words that these are words that microbiologists use that allow them to tune their behavior. And one thing that's interesting about these chemosensory arrays is that they enable bacteria like E. coli to respond to stimulus quantities over 10 orders of magnitude, enabling highly sensitive and precise responses to changing conditions. Other species of bacteria, what?
You have 10 minutes approximately for your talk left, and then questions.
Really?
Yes.
I am so sorry. I am so sorry. Boy, I really blew it, didn't I? So sorry. Okay. Yeah. All right. So with networks give rise to predictive behavior in microbial genetic networks. What they found was that they could take a normal stress response in E. coli, in which that has 2 steps. First, a rise in temperature when the bacterium enters the body, then a respiration switch from aerobic to anaerobic. They can be reversed experimentally so that 2 appears before 1 over many generations. And there are recognizable conditioning effects, such as the stress response is dissociable. It can be decoupled, suggesting a critical regulatory package, not combined responses to 2 stressors. And pre-exposure to early stimulus and temporal sequence reduces reaction time to the second stimulus. And if you only provide the early stimulus, it leads to loss of response to the second stimulus, all of which are patterns that are followed in mammals. So temporal order seems to be embedded in regulatory networks. And so they think that environmental anticipation might be ubiquitous in nature. So there's more of that, but we're just going to move on. Okay, so cell adhesion also concentrates resources and minimizes wire. It seems to be an ancient strategy. Stromatolites in Western Australia, which are 3.8 billion years old, are believed to be bacterial colonies, although this is not uncontroversial. The glue that keeps bacteria together is EPS, or extracellular polysaccharides. And the benefits include security from predators, accessing novel resources, metabolic efficiency, genetic exchange, and acquiring new competencies. In multi-species biofilms, like this biofilm on the surface of the human tongue, there, in addition to security and genetic exchange benefits, it enables exploitation of secondary metabolites secreted by non-conspecifics. One taxon's waste is another's food, but they're really quite, they're organized only insofar as proclivity, not through collective behavior. Whereas in single species biofilms, you have coordinated collective behavior, you have altruism. For example, a lot of the cells involved in moving to sporulation commit suicide. They're calling it programmed cell death, but they're not exactly sure what the program is. We know what it is in multicellular organisms, but not we haven't got a clue in bacteria. It also enables the establishment of infection or predation within taxon cell differentiation and greater structure. Now, I am going to race to the parts that are unexpected. Cell-cell communication, the principle is analog is cheapest, where necessary combined analog primitives, i.e. signaling proteins with pulsatile processing. That was true in this B subtilis biofilm we saw earlier. And it also is, okay, one of the points that I'd like to make here is that quorum sensing, which is chemical signaling in bacteria, is in logic, in functional logic. They behave like hormones, cytokines, and many neuropeptides. And every single form of chemical signaling on this chart is represented except endocrine signaling, which depends on a circulatory system. Quorum sensing systems are really important because they control social behaviors, which have a positive fitness effect on other bacteria, or whose benefit to the responding cell depends on the presence of other bacteria in the environment. I think I will just go through, I will wax through this, and we will get to extracellular vesicles, because it wasn't until fairly recently that we even knew that extracellular vesicles were used by bacteria. It was assumed that their cell walls were too tough. Pseudomonas aeruginosa was the taxon in which they were discovered. It has 5 quorum sensing systems, and it's as a opportunistic pathogen, which, for example, infects the lungs of cystic fibrosis patients. It needs to make sure that it's with its own kind, and that's what they now think these multiple quorum sensing signals might be, is just making sure we're with us and not them. Now, division of labor, I'm going to go straight to my chart, and that this relates to the neural design principle of specialization. Now, I'm not going to talk about the cyanobacterium Anabina, because now that I've listened to Detlef, I don't think it qualifies. But in Bacillus subtilis and Myxococcus xanthus, there are some things that there are quite different cell types. And I'm going to concentrate on Myxococcus xanthus here, because they have things called tan cells, and yeah, okay, both species end up with biofilms, or in the case of Myxococcus, with fruiting bodies. And so they're differentiated within the biofilm and the fruiting body. In Myxococcus xanthus, tan cells cannot swarm, which is the dominant form of predotaxis, but they seem to survive much better than the yellow cells, which are really, really adept at swarming, but have a tendency to lyse easily. Now, in the fruiting body, you have two cell types that are quite different. One is the myxospore, not an endospore, and the peripheral rods, which are they're like guard cells, actually. They're around the fruiting body, and they keep other bacteria away from them. And in both cases, there's a proportion of the population that that commits cell death. It's lysed so to provide nutrients to the next generation. Okay, I think we won't go through oscillatory activity in general, because it's been well known in yeast for a very long time. But there is a recent experiment that was really interesting, in that in multicellular animals, there are many organs that feature subpopulations of spatially differentiated and temporally synchronized cells. The question is, how do they do it? Yeast were packed into this diffusion limited microfluidic chamber to test whether a group of individually oscillating cells, which yeast are, could similarly form subpopulations of spatially differentiated and synchronized cells. They did. And what they did, this is these are photographs from the most beautiful video sequence I've ever seen. It's just, you can see the oscillations, you can see, you can see the lights going on and off. It was so compelling. And researchers, this I find interesting, researchers concluded that the interplay of intracellular biochemical reaction networks with generate the oscillations, together with quorum sensing type communication, resulted in the generation of the subpopulations of synchronized cells.
Pamela, I would ask you, I have to finish quite soon, like in 2 minutes, if it's possible, so that we have some 10 minutes for questions.
Okay, excellent. Okay, Predataxis. Okay, I'm going to combine Predataxis and Myxococcus with the idea of coordinated responses, because Myxococcus xanthus is a really badass bacterium. It is predatory, it's territorial, it doesn't like its near kin, it only likes its closest kin. And what you see here is a Myxococcus xanthus running through a population of E. coli. And what's happening here at the basement is, here are the prey cells, here's the Myxococcus xanthus running through them, and these outer membrane vesicles include, it's believed they include bacterial toxins to basically lyse the prey and degrade their biomass for acquisition. How these oscillations are generated is by the movement back and forth of cells with a dynamic protein cluster that shuttles back and forth from one end of the cell to the other, according to the direction that the cell is going in. And so they're moving helically like this, and they're also reversing direction, and that's what allows them to go very efficiently through the prey colony, generating these oscillations. Okay, so stress responses, growth responses, etc., are large-scale global responses that are triggered are triggered by transcription factors, and can involve hundreds of genes. And in this case, in fruiting body formation and xanthus, that's what they're doing. And here is a fruiting body wasteland, where there's some little bit of predataxis happening here, but pretty much all the food is gone. Okay, what happens? M. xanthus has a life cycle that you can see here that goes from individual cells through collective endeavor to sporulation to fruiting body to aggregation, and the aggregation is just amazing. It was described by one researcher as the great herd migrations of the Serengeti. And when you look up close at one of these fruiting bodies, I mean, my God, they look like a skyscraper. They're just amazing. And so what these do is after 31 hours, a mound is formed. And then after 61 hours, a fruiting body is formed, and it's fully formed after 72 hours. Now, take home messages. The 7 common mechanisms revealed by a basal cognition approach fit Sterling and Laughlin's 7 principles of neural design without undue strain. I'm not saying it's perfect, but it seems to be approximate. If correct, this means design principles that informed brain evolution already were involved in cognitive function long before nervous systems evolved. The alignment is important because it connects cognition in bacteria with cognition in animals with nervous systems and supports the notion of a continuum of cognitive function from simple to complex. And it also suggests that simpler alternatives to current dominant model systems exist, which may help to solve perhaps many problems in neuroscience. So the principles were there, nervous systems built on them. And I would like to thank my team and funding body, without whose care and support this work would not be possible. Thank you.